Next, let me explain how temperature affects enzyme activity, with the help of a graph that illustrates this relationship. To begin with, the graph shows how the rate of enzyme activity changes as the temperature increases, ranging from 0 to 60 degrees Celsius. This setup allows us to observe the correlation between temperature and enzyme activity. Initially, as the temperature increases, the rate of the reaction also rises. Why is that? Well, higher temperatures provide more kinetic energy to the molecules involved. This increased energy leads to more frequent and more forceful collisions between enzymes and their substrates. As a result, the reaction proceeds at a faster rate. However, this increase doesn't continue indefinitely. The graph shows a peak, what we refer to as the optimal temperature. This is the temperature at which the enzyme functions most efficiently. At this point, the enzyme's active site is in its ideal structural form, maximizing the catalytic activity. However, beyond this optimal point, the reaction rate starts to decrease. This is because too much heat begins to disrupt the enzyme's fragile three-dimensional structure. At high temperatures, enzymes begin to denature, their structure unravels, and the active site is distorted or destroyed. This loss of structure directly impairs the enzyme's ability to bind to substrates and catalyze reactions. So, the shape of the graph, a rise followed by a sharp decline, vividly illustrates both the sensitivity of enzymes to temperature changes and the concept of an optimal temperature range for biological reactions. This is a key reason why living organisms must carefully regulate their internal temperatures to maintain efficient enzyme function. Now, if we turn our attention to the graph, we can clearly observe how temperature influences enzyme activity. This is a classic enzyme activity curve, and it tells us quite a bit, particularly about the concepts of optimum temperature, reduced activity at lower temperatures, and denaturation at higher ones. Let's start with the optimum temperature, the peak of the curve. This is the point where enzyme activity is at its highest. Why? Because the kinetic energy of both enzymes and substrates is just right, allowing for the maximum rate of successful collisions. At this temperature, the enzyme is functioning at its most efficient level. For many human enzymes, as you can see here, this optimum is marked around 37 degrees Celsius, which aligns with normal body temperature. Now, if we look at the lower end of the scale, let's say around 10 degrees Celsius, also marked on the graph, here the enzyme activity is much slower. That's because, at lower temperatures, molecules have less kinetic energy. Collisions between enzymes and substrates become less frequent and less energetic, leading to reduced reaction rates. The enzyme structure remains intact, but the activity is significantly slowed. Then we move beyond the optimum, toward higher temperatures, like 50 degrees Celsius, which is also highlighted here. As we go past the optimal range, we see a sharp drop in enzyme activity. This is due to denaturation. At these elevated temperatures, the enzyme's active site starts to lose its specific shape, a critical feature that allows it to bind to substrates. Once the shape is altered, the enzyme can no longer perform its function effectively. This structural breakdown ultimately halts the reaction altogether. So, this graph not only provides a visual representation of enzyme activity across temperatures but also reinforces key biological principles, namely, the importance of optimal conditions for enzymatic function, and the sensitivity of enzymes to thermal changes. So, from all of this, we can conclude that temperature plays a crucial role in the rate of enzyme-controlled reactions. As temperature rises from around 0 degrees Celsius up to the enzyme's optimum, which is typically close to 37 degrees Celsius in the human body, the rate of enzyme activity increases. This is because the added heat gives molecules more kinetic energy, causing them to move faster and collide more often with substrate molecules. These increased and more forceful collisions result in faster reaction rates. In fact, for many enzymes, every 10 degrees Celsius increase can nearly double the rate of reaction, up until that optimal point. On the other hand, at lower temperatures, both enzymes and substrates have less kinetic energy, so collisions occur less frequently and less effectively. This leads to slower reactions, though the enzymes themselves remain intact, just less active. However, once the temperature rises beyond the optimum, typically between 40 degrees Celsius and 50 degrees Celsius, enzyme activity drops sharply. Even though molecules are moving faster, the excessive heat starts to damage the enzyme structure. This results in denaturation, a permanent change in the enzyme's shape that prevents it from binding to its substrate. Once an enzyme is denatured, it loses its function permanently, and the reaction comes to a stop. This entire process is often shown in a graph. The curve rises steadily with increasing temperature, peaks at the optimum, and then falls rapidly as denaturation sets in.